want you to know for the protozoans. So we're going to talk a little bit about the taxonomy, but I can tell you right now we're going to spend zero time on the phylogeny because it's basically a bush. We'll talk about some of the general characteristics of protozoans, which I'm pretty sure you already know now, and then we'll talk about some of the select groups I want you to know and their key features. So I already showed you this. This is just a copy of the phylogeny that's in the front inside cover of your book. But as you know, we're not going to cover all of these taxa. Instead, here's that phylogeny I showed you previously in class, and this is your roadmap for the entire semester, with the exception of we're really not going to cover the acanthocephala, but we'll cover those details later. What you need to know is that, so this is all of the Animalia, with the exception of this clade right here, the protozoans that we're talking about. And notice how it's the first branch off. Everything after this branch are the metazoans, are the animals, okay? So we're covering these because these are fascinating creatures, but that's not the entire reason that we're studying them. It's because they have a lot of animal-like characteristics and yet are not animals. And you know the definition now. To be an animal, you have to be multicellular, motile, heterotrophic, and a eukaryote. Well, these guys have some animal-like characteristics, but the key feature is they're not multicellular. So just to review, recall that the category of domain, seen here, so that's a taxonomic category that's above the rank of kingdom, and we didn't cover it previously when we talked about Linnaean's hierarchy. But currently we recognize three domains. The bacteria that are sort of self-understood there, so they are bacteria. The archaea, which are some previously thought to be very ancient organisms. And then there's the eukarya. And so you can see some very familiar things within the eukarya. Here's the animal, so we're in there. You got fungi, you got plants, and you got a whole lot of these other things. And if you were actually to look at some of the most recent literature on the eukarya, you would see many more phyla of protozoans than you're actually seeing here. And I think at last count there were something on the order of 22, and more are being discovered all the time and described. So you know that the eukaryotes are defined as organisms with a membrane-bound nucleus. We talked about that in class. And so just to remind you, here's a typical animal cell. It's not a plant cell. But either way, plants and animals, among other things, and fungi have membrane-bound nuclei. So, and I'll say the same here. So recall that those are different from the protozoans, so to speak, or the bacteria and what the archaea have. So they don't have membrane-bound nuclei. So these protozoans are very simple animal-like organisms, but they are not monophyletic. As you can see, they don't all descend from a single common ancestor. So that makes it kind of hard to teach. We're only going to look at a few examples, though. And just to reiterate, there's the definition of the metazoa. Okay. Now, this is a phylogeny from Chapter 5 that shows you sort of an array of the protozoans that have been described, or at least some of the groups. Fortunately, we're only going to cover a few of these. Okay, so we're going to talk about the euglenozoa, straminopiles. We're actually going to look, we're not looking at the alveolata, but we are looking at the foraminifera, the one green algae that's in the plantee, and then the amoebozoa and the pisciconta. Now, I should probably point out that we're also looking at another amoebozoid group, the Heterolobosea. It's another sort of amoeba-like thing. And we are going to look, actually, I think we're going to look at the dinoflagellates, but let's see how it goes. But I'm not going to require you to know this phylogeny at all because it's not this resolved, even though it looks this way in a tree. Recent research has it looking differently. So you should be familiar with the theory of endosymbiosis. So this is it's called a theory because, as opposed to a hypothesis, because there's been a lot of information that's been brought to bear on this particular concept. And so we currently believe that it best explains the origin of eukaryotic organisms as well as some of their organelles. So to just go over it briefly, you should read more about this in your book. At one time, there were some ancestral prokaryotic organism, single-celled. You can see it stereotyped here. What we see cells doing all the time is making these infoldings of the membrane into the cell. So we think ancestral prokaryotes were doing this. And as they did this, some of these infoldings were maintained. They didn't go away. They didn't move in and out. And they ultimately formed the endoplasmic reticulum and some of the other organelles, including probably the membrane that went around the nucleus. Because these things need to feed, just like all organisms, they were engulfing all kinds of particles or other, say, prokaryotic organisms. And so there are aerobic and anaerobic examples of these early prokaryotes. So again, living in oxygen-rich or oxygen-poor environments. And so what we believe happened is that there was an engulfing of an aerobic or oxygen inhabiting prokaryote. After it was engulfed, it was basically, instead of dying and being digested by the prokaryote, it was maintained and ultimately formed what we now know of as the mitochondrion, so the powerhouse of the cell. In addition, there were some that also engulfed some photosynthetic prokaryotes, and these ultimately ended up forming the plastids or the chloroplasts in eukaryotic cells. So we think this is how some of the organelles arose in the eukaryotic organisms. Okay, let's go back to some of the general characteristics of protozoans. So there's a box like this in your book. You should become familiar with it and know pretty much all of these major characters, but let me point out several of them. Let's walk through them pretty quickly. So the first one says they're unicellular. That's easy, but remember that some of these live singly like this, but some of them actually form colonies where they kind of hang out together or are attached to each other, like you see here. Now, when they're attached to each other, but they're formed of single-celled organisms, it's still considered not multicellular. So we call that colonial. So they're still independent cells. They just happen to be living together, but they don't have to live together. So they're mostly microscopic. You can't see these things with the unaided eye. They represent all kinds of body symmetries. They could be radially symmetric. They could be spherical. They could be asymmetric, bilaterally symmetric, you name it. Keep in mind that these have no germ layer. So these guys do not derive from endoderm, ectoderm, mesoderm. They don't have any of those layers. They, being a single-celled organism, means that they, by definition, do not have organs, but they do have structures inside them referred to as organelles that perform many of those functions. Remember what the definition of an organ is and what a tissue is, right? In order to form tissue, you have to have cells of different types that are working together for a particular function. And when you have multiple tissue types working together for a particular function, that forms an organ. These things aren't like that. Go check it out in your book. I think I have a cell or a figure to show this. So they can locomote in a variety of different ways. They can use cilia, like you see, here we go, all around this particular protozoan. You could have, I might be going ahead of myself, let's see, here we go. Many protozoans, and you're going to find that animals throughout this semester form what are called symbiotic relationships. So they have some kind of ecological relationship with another organism that could be good, it could be bad, it could be indifferent. So to go over some of the symbioses that we'll see over the semester, they can be mutualist, so engage in a mutualism, and we indicate that as positive-positive, meaning for the two organisms that are interacting, it's mutually beneficial. They're both gaining a benefit. So one can't really live without the other. And so an example of that you're probably familiar with are sea anemones and clownfish. So they both gain because the sea anemone has a home that's protected. Sorry, the clownfish has a home that's protected by the sea anemone, right? Because the sea anemone has stinging tentacles, and so no other animals can really bother the clownfish because it can safely live within the tentacles without being stung. And at the same time, the clownfish will often either chase off other organisms or it can provide nutrients to the sea anemone. So they both gain. you can have a symbiotic relationship that's referred to as parasitism. So that's positive for the parasite. It's negative for the organism that's being parasitized. So you're removing resources from one organism to another. And a classic case of this is not the mosquito per se, although it is a parasite, but it's the plasmodium that you're going to learn about, the organism inside the inside the mosquito that is the true parasite and is taking resources from its host, often humans or other mammals. And then one you may not be familiar with is called commensalism. So this is where one organism benefits from the relationship. The other one doesn't really seem to be negatively or positively affected. It's just kind of there. So one example that often comes up in the literature, although this is debated, is with barnacles that are living on whales. And so you can actually see a few of the barnacles here and a few like right here. So apparently these have virtually no impact to the whale. It doesn't seem to slow them down. It's not really taking resources from them. Like, you know, they don't seem to scratch them off. So unless you have a high load, it's really not bothersome at all. And most of them don't have a high load. It's beneficial to the barnacles because they're able to encounter more food resources and they're better able to feed than those that are actually hanging out on rocks on the bottom of the ocean floor and have to compete with other barnacles for food resources. Okay, let's go through some more of these features. So they can locomote with a number of structures. So we already saw cilia. You're familiar with flagella. They could use pseudopodia like you see in a number of these organisms here. And these are a number of different sort of amoeboid-like organisms. Sometimes their pseudopodia look very sharp and kind of sticky and needle-like. So pseudopodia, by the way, are defined as temporary protrusions of cytoplasm that function in locomotion or in ingestion. Okay, I don't know if I can actually make, I might be able to make this link work. Look at that. So to show you a video of this, not sure how well this is going to work, but we're going to find out. Okay, no offense, but I think you get it, so we're going to turn that off. Okay. So, as I mentioned, I don't think I did mention this to everyone in class, just to a few people. So, there is no morphological difference between a flagella and a cilia. So, they all look like this, and they have what's referred to as a 9+2 construction in their structures. And so that refers to the number of microtubules that are inside either the cilia or the flagellum. And they're showing you a diagrammatic representation of it here. By the way, this is a pet peeve for me because they come in pairs. You can see them here, 2, 2, 2. So there's 9 pairs around a single pair in the center. So since they're talking about it as being 9+2, seems to be it ought to be either 9+1, because we're only talking about pairs, or you should actually say it distinctly. It's 9 pairs around a single pair in the center. Anyway, just my pet peeve. Here's a video of flagella movement. You guys will have already seen this in lab. You can see 2 flagella right here. So this is a Chlamydomonas, a kind of green algae, by the way. Oh. All right, we're going to speed this up. Oh, there you go. That's got 8. Somebody was asking me what's the most number of flagellae. There you go. Here's cilia all around. Okay. So let me tell you about the difference between cilia and flagella. It has everything to do. Let me make sure I don't have this on the next slide. I do. Okay. So the difference between a cilium and a flagellum has to do with the movement of the water relative to the structure itself. Okay. So a flagellum, if this is your flagellum, water is going to flow past, or the fluid's going to flow past, parallel to the actual structure of the flagellum. With a cilium, because often you have them like this, where they're completely surrounding the organism, water is going to flow perpendicular past the cilium. Okay? So it's always going to flow in the same direction around the body. It's always going to go parallel to the body. But what makes the difference between a flagellum and a cilium is the fluid's flowing parallel to the flagellum, perpendicular to the cilium. All right, what else we got? Oh, yes. So you have all kinds of feeding strategies throughout the protozoans. You have autotrophs, those that make their own food through photosynthesis. So if I throw up a picture and you see green structures inside, those are probably chloroplasts, and it's a sure bet that they can photosynthesize to feed themselves. Heterotrophs, by definition, ingest food items, and they can be all kinds. They can be liquid, they can be solid, they can be all kinds of things, but they ingest their food. Here's another quick video. Don't think this really shows you much. Actually, and you gotta love some exciting music to go with your protozoans feeding. Anyway, not that great, showing you a variety of protozoans. Let's speed this up. Okay, so when you talk about heterotrophy, there's a variety of different types. So phagocytosis is when you're consuming solid, that should be food items, not foot items, but I suppose you could ingest feet. I'll show you this other movie in a minute. Penocytosis is when you consume fluids through modifications of the pseudopodia. And just so you know, here are different kinds of amoeba types, and here's an amoeba eating a Chlamydomonas. Here's one attacking a paramecium, and here's two attacking a paramecium, and so on. I don't think this is my amoeba movie, but if it is, it's worth watching. Let's see. Oh yeah, this is awesome. It's hard not to get excited about amoeba when they're feeding. Oh, look at that. Here it goes. All right, so it's going to engulf this Chlamydomonas right here. It's putting out its pseudopods. Poor unsuspecting algae has no idea what's about to happen. It's happy. No idea, no idea. And the pseudopodia close up. Uh-oh, uh-oh. It's about to realize. Watch. Now it's figured it out. Poor algae. Okay, and you can virtually hear the amoeba burping. Right. Going back. Okay, so when it comes to reproduction of protozoans, and I said we were going to be talking about reproduction a lot over the course of the semester. So you can reproduce asexually or sexually. Asexual reproduction is defined as producing offspring without genetic recombination. And so you're generally producing clones. And so there's a number of protozoans that'll do this, and they can do it in a number of ways. They can engage in what's called fission. So in this case, it's a process of reproduction via cell multiplication where offspring and adult are roughly the same size. And basically, you can think of it as taking an organism and splitting it down the middle. You're going to see some animals that can do this too. And you can either split in half, either anterior or posterior. You can do it in half, but basically it's splitting apart into two, and you form two organisms that are the same size. So this is just an example. So you start out here with an adult. It basically goes through a series of divisions, and you have two offspring that are roughly the same size. Budding is a similar yet different process. So it's still a reproduction via cell multiplication, but what happens is a little part of you sort of pooches off and buds off and then separates, but now your offspring is smaller than you. So you can see this happening here. So here's the adult, and here's actually 2, possibly 3, maybe even 4 offspring that are budding off. And the offspring will bud off initially smaller than the adult, and then they will grow. So just to review a couple of these topics in terms of digestion, lots of different ways to get your food or your nutrients for life, autotrophic, heterotrophy, and there's a couple of versions of heterotrophy. In terms of the excretory system, so we didn't actually cover that. How do they get rid of waste? Well, normally they're using either contractile vacuoles or food vacuoles to deal with various forms of waste, but also contractile vacuoles are used to control water balance. In terms of locomotion, you can do it via cilia, flagella, or pseudopodia, and in some cases protozoans are basically just flowing with the current, the fluid in which they live in. They have no nervous system. So these guys are getting everything done without a nervous system, which is pretty amazing. For respiration and osmoregulation is water balance. Respiration is, of course, oxygen balance or gas exchange is a better term. So this is basically handled by contractile vacuoles. It's going just straight through the membrane. They do not have a circulatory system, so we're dealing with organisms that are very simple. We talked about reproduction. They can do it asexually or sexually. I didn't actually cover conjugation, and we're not going to cover the details. What's important to note here, if you take micro, you'll get this in detail. What's important to note is that you start with 2 adult individuals. They are going to exchange genetic material, but unlike how it happens, let's say, something you can relate to with humans, you start out with 2 adults, their gametes come together and form a single individual, right? So it's just 2 people making one organism at a time anyway. What happens with these guys is as they exchange genetic material, they go through a series of processes and they end up, in fact, with 8 individuals rather than just the 1. Now, let's skip these questions. All right, let's talk about the individual groups and what makes them unique. So in the phylum Straminopiles, also known as the Heteroconta, and by the way, this is a term that needs defining. This term, conta or cont, that refers to, so that's Greek, and means either tail or better yet, it means pole or tail. And in this case, it's referring to the flagellum. So it's a tailed or flagellar organism, and hetero means different. So it has different kinds of flagella. Now don't be confused. The example that I'm showing you here has 2, but that's not necessarily the defining feature of all members of the Straminopiles. Okay? They do have different kinds of flagella, but it doesn't necessarily mean 2. They could have 4 flagella that differ. Some are short and some are long, some are hairy, some are smooth, right? They could have 6, they could have 8. So it doesn't refer to 2, it just refers to having more than 1 flagella and they're different from each other. Now here I say have 2 different flagella, but that's just in this example that I'm showing you. So they have they have 2 or more different flagella. So what you can see here is this one is meant to be hairy, this one is smooth. They call it a tensile flagella. In particular, though, these flagella are inserted in the anterior end. And I know this looks funny because you're like, well, the anterior should be here. Well, wherever these are inserted, that's the anterior. And they're usually moving in the direction of where the attachment is. So they're moving this way. One is long, one is hairy. Usually one is long and hairy, the other is short and smooth. But they can have more. So it doesn't necessarily mean 2, it just refers to that they're different. So there's a number of algae that actually have this, these key features, among others. There's all kinds of species within the Straminopiles, but some of them look quite plant-like. In this case, what you're seeing as the example of the Straminopiles is this yellow scudge here. So it kind of looks like scudge, but if you looked at it under a microscope, you'd see something that looks like this. And then some of them have exterior skeletons called tests, and they are really variable, and they look as you see in this picture here. Oh, by the way, these are diatoms. These guys are diatoms. So you hear about diatoms being used. These are actually quite important to us ecologically So diatoms are usually a great example or a great way to determine if you have clean water sources or polluted water sources. If you have diatoms in your water and there's a great diversity of them, you've got really clean water. But if you have no diatoms at all or the diversity is low, then there's something wrong with your water source, or if there was previously a high diversity. So these are usually a great indication of water quality. So the phylum Viridiplantae is where all the plants are usually found, but it's also where some green algae are classified into. And basically you can think of green algae as being not necessarily the precursors, but they share a common ancestor with the plants. So they have chloroplasts, they can photosynthesize, and the examples that you would have seen in class probably include Volvox. I don't think you guys saw Gonium in lab this year. So they are characterized as being either solitary or colonial. I'll give you an example, here you go. Some move via flagella, but there's a few that don't have flagella. Here's an example of Volvox. But they do all have chlorophyll A and B that they use to photosynthesize with. So here's a single Chlamydomonas, and you can see its flagella here. This is Volvox, and you have the mother colony, and inside are the smaller, what are referred to as daughter colonies. So each of these little dots is a single individual, and they live colonially. And this is an example of the cycle, and I don't need you to know the gory details of the reproductive cycle, but what I do want you to know is that they can reproduce both asexually and sexually. The sexual part is pretty straightforward. It's not unlike us. There are male and female individuals. The males produce sperm. They pass it over to the females, usually just by transmitting it in the water, and it flows over to the female. And then the female has eggs that can be fertilized and produce more individuals. In the case of the asexual, what happens is she does this infolding, and there are these independent cells that'll just peel off from every individual and form these daughter colonies that will form together. The second phylum that you're going to look at is the Euglenozoa. So this includes two genera that we're going to look at, Euglena and Trypanosoma. So these guys are characterized as having what's called discoid mitochondrial cristae. So you know what your mitochondria looks like, right? It's kind of, they show it to you as being kind of football shaped, and it's got a little squiggly line in the middle, or that's what it often looks like. So kind of like that. Theirs is discoid. It looks like this, so it's kind of funky. It's got these little circles all over it. They're also characterized as having longitudinal microtubules beneath the plasma membrane that form the pellicle. So you guys, lots of you were looking at this in class. So they have these microtubules that form this sort of spiral shape, if you will, within the membrane, and it gives rigidity to the body structure and gives it some strength. So this example, Euglena has two heterocont flagella. So it's got one short one and it's got one long one, but they're otherwise both smooth. And some of them have a stigma, here you go, otherwise known as an eye spot, so it allows it to sense light and dark. Now, why do you think that's important to do? If these guys are photosynthetic, they probably want to feed by moving towards the light, right? So they need to be able to figure out where the light is. Not a good idea to be swimming down to the bottom of your habitat where you probably can't reach the light. So trypanosoma is, of course, medically important to us. It causes, among other things, African sleeping sickness and Chagas disease, depending on which one you're talking about. These guys are characterized by having one or two flagella, and so you're actually seeing one here and one here in this example. The mitochondrion extends throughout the body, but you can't see that in this picture, so they're not actually showing you the mitochondria in this picture. All of them within this genus are parasitic. And so they can cause us great problems as humans when they invade our bodies. They can make us very sick, or they can kill us. So it's very important to understand protozoans and the ones that can make us sick. And so for those of you going into the medical field, you need to know a little something about these and be able to identify them so that you can help your patients. The third is the phylum Apicomplexa. So there's two genera that you probably would have seen in lab this week, Toxoplasma and Plasmodium. So these guys are all parasitic, they have what's called an apical complex, and if I show it to you here, this is the apical complex. And without going into the gory details, what this apical complex does is it allows it to bore into other cells and invade, and then reproduce once they've gotten inside of the cells that they're trying to enter. So these are quite damaging in terms of as a disease-carrying or as a disease agent. They're not only characterized by having this apical complex, but, I should probably say, notice that they don't really have any locomotory organelles, and they don't have a flagellum, they don't have cilia. Sometimes they have pseudopodia, but often they're just flowing through a cell or through the environment just with the current. But I should say when they, cilia and flagella are generally absent. So, parasitic, have an apical complex, lack locomotory organelles. I don't really care that you know the Plasmodium life cycle. If you want to know more about that, you can take microbiology. But it's important to note that Plasmodium is in the apicomplexa and that they are transmitted by mosquitoes, and it's always the female mosquitoes that are feeding, not the males. Okay. The next group is the phylum Ciliophora. So the name should probably tell you everything, covered in cilia, right? So they have cilia at least at some point in their life. They're often arranged in rows. You can see some examples here. You got a row of them here. It almost looks like a mohawk somehow. Lots of rows here. This one just has a crown of cilia at the top. So they've got cilia somewhere and at some point in their life. They're often arranged in rows. They have trichocysts or toxocysts that are used for defense or capturing prey. So these are structures that they can basically harpoon out to try to defend themselves or to try to capture prey. And they usually have two types of nuclei, so a macro and a micro nucleus, and sometimes they have multiple versions of them, so they almost look like a chain of pearls. But sometimes they just have two. So these are some examples of some trichocysts, and they can look like quite long harpoons that can be quite damaging to their prey. Now these are too small to really hurt us, but they could. And I think in lab you probably only saw paramecium and maybe stentor. I don't think you saw these other two as examples. And there's of course a classic example of a paramecium. So the macronucleus is responsible for the metabolic and developmental functions. The micronucleus participates in sexual reproduction and gives rise to macronuclei afterwards. We're not going to go into the gory details of this. You can get that later in a subsequent class. But you do need to know the difference between the macro and the micronucleus and who's got them. Okay, the dinoflagellata are very important to know. So these guys are characterized by two types of flagellae, what are called a transverse and a trailing. So just to give you an example of this, here's Seriaceum, and there's quite a bit of diversity in the dinoflagellates. But in this case, it has this groove. I think of it as a funky Death Star. It's like a spiky Death Star that's got the little groove around its middle. And there's a flagellum that comes out of the groove, and then there's usually another flagellum that comes out the top or the back. So there's a transverse flagella, and then there's a trailing one that comes out, maybe the equivalent of the posterior end of the particular individual. And some, but not all of them, are bioluminescent. So if you actually go into the ocean where these things occur, if you actually wave your hands or your feet around, you can actually see them flashing their molecules that make them bioluminescent. They're not everywhere, but they're generally salt water. And that I have a link for this, but it's really hard to see. So if you just Google up bioluminescent dinoflagellates, you'll find all kinds of really cool videos. And so it's these guys, the dinoflagellates, that are responsible for the red tides. So the red tide, thought I had a few more slides in there for you. So the red tides are dinoflagellates, and what's happening is it's not necessarily the dinoflagellate itself that's making you ill. They're producing a toxin that makes you ill. What they're doing is the red tides are these spawning periods where they're reproducing in great numbers, and it's usually due to an influx of nutrients. Not always. It can be due to other things too, but in any case, when they are in great abundance like this, they're often releasing pretty heavy-duty toxins. Those toxins are taken up by things like oysters and mussels and filter-feeding organisms. And if you eat those organisms, when they filter fed on these red tides, they can kill you. So that's why they say it's important when you're eating things like oysters, you should eat them only in certain months of the year and not in others. Probably a better way to control that is to maybe eat them only when they're farm produced, so that they're probably not going to have any of these toxins at all at any time of the year. So this is another example of how these protozoans relate to you. If you understand a little bit about their biology and know that you shouldn't eat during certain months of the year and other months of the year it's safe to eat them, you'll have a better way of relating to keeping yourself healthy, and if you're going to be a doctor, keeping your patients healthy. Okay. So I wrote amoebas here, and this is an actual term. I didn't misspell it. If you're using the generic term for things that are amoeba-like, that have pseudopodia of any kind, that's how the term is spelled. But as it happens, there's two independently derived groups. These guys are not closely related at all evolutionarily: the Heterolobosea and the Amoebozoa. And so these are mostly free-living organisms that move via pseudopodia. The pseudopodia can look differently. They can be needle-like, or they can be sort of lobular-like. But they all move in kind of the same way. They may or may not have a test. This is free-living. These guys have like an exoskeleton on them. They can be, they can either be engaged in phagocytosis or pinocytosis. So they can be quite variable, but these two groups are not closely related. So this is a classic example of what's called convergent evolution, when you have features or maybe even whole organisms that look incredibly similar to each other, but they don't share a common ancestor. They're completely independently derived. And the reason that happens is that generally they're living in environments or have some sort of selective pressure on them that has moved them, or have selected for certain features to be most beneficial in that environment, and those features are the same. So they're similar environments, but in totally unique and separate places, and that results in them looking very, very similar. So there are organisms, for instance, that live in the desert southwest of the United States, and also in deserts in Australia. And the example I'm thinking of has to do with lizards. And because they live in dry, sandy environments that have sort of similarly colored sand, and the only place to live is in these little rock outcrops, they look incredibly similar to each other. So they're sandy colored, they're sort of spiky, they move in a similar way, and they actually eat the same things that eat ants. And so there are lizards that look almost identical to each other because of the environments that they live in, but they're not closely related at all. Okay, last one, yay! So the phylum Epistheconta is basically everything else. So we've covered all the protozoan groups, but everything else of the fungi, we're not going to talk so much about the choanoflagellates, but then all the other animals are in the Epistheconta. So remember, conta means tail or pole, and it refers to a flagellum. Opistho, also derived from the Greek, means basically anterior. So, posterior means, no, it's anterior, means anterior. So that means at some point there's a flagellum at the anterior portion of the body. So here we go. Here's bacteria, archaea, here's the eukarya, all of this. We covered some of these guys. And here's the Epistheconta. Here's where we're going to go for the rest of the semester. Oh, posterior. It is posterior. Okay. So a second ago I said anterior. It's posterior. That's right, because if you think about sperm, it's on the back end. Yeah, duh. Anyway, so opistho means posterior flagellum. And they have flattened mitochondrial cristae, like with our typical sort of mitochondrial. So like ours, the football shape. So we're not going to go into it now. We're going to talk more about metazoans next time. Here's that phylogeny again. So now we've covered the protozoans. Next time we're going to cover the porphyrians, or the sponges. And actually, you don't need to know this part. So that's it for this podcast. I hope this is helpful to you. And if I can answer any other questions, email them to me or talk to me in class. Check you later.